So our next speaker is Dr. Liu from the National Cancer Institute. She's going to be talking to us about single-cell barcoded transcription factors to study plasticity in neuroblastoma.
Thank you for the opportunity to present our work today. I'm postdoc fellow from Dr. Carosel's lab. Today, I'll be talking about how we identify the transcription factors that regulate neuroblastoma plasticity and drug resistance. As we've heard from Dr. Yermaro, she already gave a nicely introduction of neuroblastoma. So I'm going to focus on neuroblastoma cellular plasticity. Neuroblastoma derived from multipotent neurocluster cells during differentiation and exhibit significantly heterogeneous. Two major cell states have been defined. One is called the adrenergic, which is more differentiated, proliferative, and chemoresistant. The other is called the mesenchymal, which is less differentiated, stress-adaptive, and therapy-resistant. This bar chart shows how tumor compositions evolve under different treatment. At diagnosis, most tumors are adrenergic dominant. After chemotherapy, there is a persistent population which is associated with chemoresistance and eventually drive relapse. So this raises an important question to us. What controls these cell states' dynamic and reversible transition between each other? To answer this question, one possibility mechanism is that transcription factors may control these transitions. We know that transcription factors are master regulators that define cell fate. For example, the adrenergic state is dependent on the regulators like FOX2B, HAN2, and GAS3, while the mesenchymal states are based on the regulator PRX1, NOTCH2, and MAMO2. We already know that PRX1 and NOTCH3 can reprogram adrenergic cells into a mesenchymal state. Our question is, what are the other transcription factors beyond these two can contribute to this reversible transition? So to answer this question, we build a barcode-based transcription factor screen approach. First, we select transcription factors from mesenchymal and adrenergic signature genes. We generate two transcription factor libraries, each one linked with a unique barcode. We packaged these libraries into neuroblastoma cells. So specifically, we introduced mesenchymal transcription factors into adrenergic type SO5Y cells. And we introduced adrenergic transcription factor library into mesenchymal type SHAP cells. We introduced the transcription factor overexpression for 7 days and subject the cells to etoposide treatment. After 17 days, we use a single cell and sequencing to capture barcode linked cell identity transition. And after 21 days, we use genomic sequencing to capture barcode linked chemoresistance. I'm going to focus on the adrenergic to mesenchymal transition today. So first, we asked what transcription factors control cell state transition. Under normal conditions, most SY5Y cells remains in an adrenergic-like state. After overexpress with mesenchymal transcription factors, we see a clear shift toward the mesenchymal-like population. And after we treat with etoposide, we see the mesenchymal population was even more enriched. This suggests that chemotherapy is not only selecting for resistant cells, but also reshaping tumor cell identity. To understand the individual transcription factors contribute to this transition, we analyze the lineage state programs associated at the single cell level. Under normal conditions, Most of the cells are clustered within the adrenergic region. However, after etoposide treatment, all the transcription factors shift the cells toward a mesenchymal-like state. Among these transcription factors, PRX1 is a known mesenchymal driver which validates our screen. Also, beyond that, TBX18 and 61 emerged as the top transition drivers in this transition. We next asked what are the contributions of transcription factors to the chemo treatment. We use the relative enrichment of DNA barcode read counts following etoposide treatment. Transcription factors that increase chemo resistance are clustered on the right side. Transcription factors that sensitize cells are displayed on the blue side. Once again, PRX1 enriched in the top hits, which validates our screen. Beyond that, 61 emerged as the top hit for the chemo resistant. This shows that 61 may have a major role in cell state transition and chemo resistance. So we then focus on 61. 61 is a homeobox transcription factors family, which plays an important role in the normal development and differentiation. 61 is known to interact with EYA protein to regulate downstream gene expression. We next look at how 61 is associated with cell state identity. We look at 3 different R2 databases, and we found that 61 is highly expressed in mesenchymal identity at the tumor transcriptional level. And in 3 different pairs of neuroblastoma cell lines, 61 protein level is highly expressed in mesenchymal-like cells. However, the EYA protein level is not specific in either cell state. We then focus on 61 in our study, so we first overexpressed 61 in different neuroblastoma cell lines. What we observed was a consistent pattern. After 14 days induction, the mesenchymal marker FN1 was upregulated. The adrenergic marker DRQ1 and DBH were reduced. At the transcriptional level, 61 active mesenchymal signatures will surprise adrenergic signatures. We next asked whether 61 reprogramming cells contribute to chemo resistance. On the right, when we overexpress 61 in different cells, we see a clear IC50 value shift, shows the cells are resistant to etoposide treatment. When we knock down 61 in mesenchymal type cells, we see a clear reduction of the cell viability, which show the cells are more sensitized to chemo treatment. We then asked whether this pathway can be therapeutically targeted. We know that 61 works with EYA together to form a transcriptional complex, regulate gene expression. Importantly, this interaction can be disrupted by a small molecule inhibitor. When we combine this small molecule with etoposide treatment, we see the reduction of cell viability in both 61-driven resistant adrenergic models and in our mesenchymal baseline models. This shows that 61 EYA complex is druggable. So to summarize, neuroblastoma exhibit 2 distinct therapeutic sensitivity and therapeutic resistant state. We built a pooled barcode transcription factor screen, identified that 61 can reprogram the cells from adrenergic to mesenchymal. Functionally, 61 drive a shift from adrenergic to mesenchymal state and promote chemo resistance. therapeutically, 6-1 maybe a 6-1 EYE complex can be targeted. Moving forward, we are going to explore how 6-1 drives neuroblastoma plasticity and chemo-resistant in vivo, and investigate whether the combination of inhibitor and chemo treatment is much more effective in vivo. And with that, I would like to thank my mentor, Dr. Xu. Thank you everyone who contribute to this work, and I'll be happy to take any questions. Thank you.
For the great talk. Any questions for Dr. Liu? I'll probably start with one while people are thinking about questions. Do you think 6-1 also drives similar plasticity in other adrenergic tumors, adults or pediatrics?
Yeah, that's a good question. There are these. Yeah, 6-1 is a master transcription factor, can drive the reprogramming in rhabdomyosarcoma.
And do you think this mechanism is specific for a class of chemotherapeutic agents or might be broad against any chemotherapy? Do you think this mechanism is driving resistance against a specific class of chemotherapeutic agents, or it would also be applicable to other chemotherapeutic agents?
Yeah, that's a great question. So we think our mechanism shows that the therapeutic target is based on the 6-1 driven resistance model. But we still have a lot of ongoing work to show whether there is a relationship between the 6-1 expression levels and the drug response. And I think, yes, the mechanism is related to the 6-1 level.
Any other questions for Dr. Liu?
Expect this with any type of treatment or with chemotherapy specifically? Sorry.
Mind repeating the question?
Yeah, so
I can repeat it.
Yeah.
So the question about transition of adrenergic to mesenchymal, if that's something you expect with any type of treatment, like for example, if you had CAR T cells or NK cells, or is it specific to chemotherapy?
Thank you for this great question. Under our treatment, we only focus on the ETOP cell, but we should probably test the other chemo like doxorubicin, the other chemo drugs, and then we may compare whether the transition was consistent. But from our results, we think that the transition is consistent across different chemo drugs. But when we if you are talking about the other like CAR T, we think the answer I may probably don't know, but I think the resistant mechanism may be consistent in these models.